phenotype level, and also at the molecular level with things like GWAS and even biochemistry to figure out mechanisms that control this increased variance in response to environmental stress. But something that I think is really lacking or is missing in these models is accounting for development. And so my lab really works specifically on the molecular and cell type specific sources of this increased variability after environmental stress. So just to give you an idea of what I mean by this, so these are zebrafish. These are normal ones growing at their normal favored temperature. They're about 2 days old. And yeah, is this a pointer? Yes, wonderful. So, you know, it's a fish. There's eyes. This is the yolk. Here's the tail here. You can see there these melanocytes. Now, if you raise these embryos a little bit higher, not a full-on heat shock, just warm, well within their range, you start to see these characteristic phenotypes. So in this case, I'm highlighting one, which is this bent tail. I'm sure you can see it. Others have highlighted many other phenotypes. And this is really characteristic of the environmental stress. And so the other thing that's really important to know is it also increases, high temperature increases developmental rate. So the hallmarks of this increased temperature during development are this increased developmental rate and the fact that this phenotype, like this bent tail, is highly variable. So again, I'm showing you bent individuals, but in any one population, you'll have those that are more severely bent than others. So these phenotypes are variable, but they're consistent. You always see the bent tail, which leads to the idea of maybe specific cell types or processes or regulatory networks are breaking down to produce these phenotypes. So, which is, you know, this is exactly how I landed on single cell as a field because I felt it was the right tool to address this question. However, typical single cell workflows, despite their many cells that you can capture, do not typically capture individual level variation or biological variability between individuals. So to go forward on this, we needed to modify some of the traditional methods. And I won't belabor this too much, but I'll just talk to you about the basic detail of how this works. So for this single cell workflow, we actually put individual zebrafish embryos in wells of a 96 well plate and dissociate those embryos. And upon kind of lysing the cells and bursting the nuclei, we add these single-stranded hash oligos, you know, which have a barcode that tells you what well that fish was in, and also gets picked up along with the rest of the transcriptome with this little poly-A sequence. So the approach we use is not the microfluidics-based like 10x approach, but rather this combinatorial indexing. And that's not so important other than it's compatible with this hashing technique and you can do very large scale experiments. Okay, so the result of this whole workflow is that you get transcripts that have a barcode telling you what cell they came from, and you have an additional barcode telling you what embryo the cell came from. Okay, hopefully that's clear. So you can encode things like treatment or whatever, but also individual embryos. So I use this technique to make a kind of atlas of temperature stress during early zebrafish development. So there are a few different time points in the early stages of organogenesis. And in this data set, 3 increasing temperatures that, again, are below the level of heat shock, but are increasingly stressful to the fish. And in this experiment, there were about 300 individual embryos and about 500,000 total cells. So I'll talk about next how we annotated these cells, because I think it's useful and relevant for this crowd. So we actually made another data set with a constant collaborator of mine, Lauren Saunders. And this data set actually has about 1,000 embryos, and it covers an even wider span of embryogenesis under the normal developmental conditions. So this is about 1 million cells, and it's covers many time points. And so we sort of painstakingly annotated this data set and wanted to rapidly transmit those annotations to our temperature data. So have used an approach that I think is becoming more and more common in this field. Sorry, come a little closer to the mic. So these are some of the annotations I mentioned, and this is what the fish look like at these stages. And what we did to transfer the annotations is just from this giant reference data set, actually just extract the PCA model and the UMAP model and use just, you know, maybe you're familiar with the UMAP transform functions or things like this. But anyway, we transformed the query data based on the model from the reference data. And in that way, we can visualize the data in the same reference space. That's why the UMaps look the same here. And it also allowed us to very quickly transfer the labels of all of the cell types that were painstakingly annotated here onto the brand new temperature data set. So just going back to this one more time, I really want to hammer home that underneath this UMAP, there are like 1,000 individual UMAPs representing each individual embryo. That's what's new about this kind of data. So what that allows us to do is make a very useful abstraction of the typical single cell gene expression matrix. As opposed to just looking at gene by cells, we can now group all the cells that came from one embryo, like this red one here or this green one here, and count up all the cells of each type and make a new matrix, which is the cell type by embryo matrix, that gives us information about the composition or the anatomy of each embryo in the data set. And this, of course, changes as a function of time and presumably with perturbations like temperature. And one last technical note, in case anyone's interested, this approach samples about 10 to 20% of the whole embryo at this stage. So that's about 2,000 to 5,000 cells per embryo. So what kinds of questions can we ask with this embryo or this individual embryo level data? So in some ways, we can just treat it just like we do the single cell data and process it in exactly the same way and look at this dimensionality reduction plot. And what we see when we do this, now each point is a single embryo, not a single cell, and you know, points are close if they have similar composition. What you see is this kind of trajectory that emerges, and that you can see is defined by the sample time point. So the time point in which these individual zebrafish embryos were sampled. And we can already address one of the main influences of temperature on this data by seeing where the high temperature embryos land. So now if we take this kind of developmental stage trajectory here and then highlight embryos that were raised at different temperatures, we can see that at one time point, one day into development, the high temperature embryos are always creeping up a little bit ahead of the normal temperature or the reference temperature. And this is true no matter what time point you sample these embryos, the high temperature ones are always ahead. And this may seem trivial. We already knew temperature increased developmental rate. However, it's not trivial to actually find a comparison, the proper embryo to compare your sample to across, you know, these different treatments. So you can kind of see almost by eye here that a 30 HPF embryo raised at high temperature would be better served by comparing to the 36 HPF control, because, you know, this high temperature increases developmental rate by about 4 hours. And so when we make comparisons of gene expression or of cell composition, this is probably the better comparison for the sample, not its matched control at this time point. So hopefully that makes sense. Temperature distorts the relationship between clock time and developmental stage. So something that's not common to do, something that's a little bit different, is we can also ask the same question at the cell type level. So we can ask, do cell types actually accelerate equivalently? So we know the whole embryo is accelerated. But now what if we look at trajectories of, say, the muscle cells? So this is kind of the reference, the full developmental reference, but of just muscle cells. And we would assume that, you know, any given embryo at 28 degrees would land kind of earlier on average in this trajectory than the higher temperature embryos. And so we can quantify this fairly easily with just a nearest neighbor averaging of all of the time point labels associated with each reference cell that neighbors one of these high temperature cells or cells from a high temperature embryo. And so in that way, you can compute a kind of mean cell type age for each one of these embryos, because of course we have many, many embryos per sample. And so when you do that, you can, so if we take just now embryos that were sampled at 24 HPF and look at across all these cell types, what is their mean age in comparison to the reference, we see that, yeah, it's close to 24 hours. And there's some variability around that. Now, when we look at the high temperature embryos, we can see that this, and these are ordered, by the way, by their relative acceleration or increase in age relative to the 28 degree temperature. We see that there's actually even variability in the degree of acceleration. So all of these guys are accelerated quite a lot, where many of the cell types on the right side here are not accelerated very much. So this kind of acceleration we see is not uniform across cell types. So this is already some, a potential source of variability in these embryos. So we can do a similar analysis to ask even further a question about timing. So with these mean cell type ages for each embryo, we can actually ask if they're synchronous or if development is proceeding synchronously. And the way I've done that is just for each individual embryo to take the mean cell type age of, say, notochord cells and compare it in that same embryo to the age of muscle cells. And the assumption is that, if development is coordinated, all of these cell types kind of traverse their trajectories in unison, if that makes sense. And so in looking across embryos at different time points, we should be able to see these cell types accelerate synchronously. And so if you look at just pairwise correlations, like in the schematic, across all cell types, this is exactly what you see. So there's, for the most part, a positive correlation, which makes good sense, and development ought to be coordinated. However, in the high temperature embryos, if we just keep the order the same on this plot, you can see that some of this correlation structure is lost. And you can just look at the distribution of these correlations across all these pairwise correlations in the different samples and see that at high temperature, there's a marked loss of basically synchronous developmental timing. So this is another kind of unmeasured or not previously appreciated effect of temperature stress during development, which is it introduces a kind of asynchrony in timing. Okay, so what else can we do with this data that's this sort of individual embryo data? So we can now start to phenotype the embryo, not just in terms of timing or gene expression, but in terms of its composition or anatomy. So one obvious question to ask is, for embryos raised at high temperature, are there any cell types that are more or less abundant? So to do that, use beta binomial regression to model both changes in the mean of specific cell types in response to high temperature, but also in the variability. So just as a reminder, when you raise these fish at high temperature, many of them look normal. So this, just to give you an idea of what a normal looking fish looks like, but a lot of them have this bend in them. So there's this kind of phenotypic variability, and we want to know if we can see that in the data. And also whether these high temperature embryos are really just like a wild type embryo 4 hours ahead, or if they have some persistent effect on their anatomy or composition. So this is a heat map showing on the y-axis here all of the different cell types in the embryo, and here the kind of developmental stage. These are 4 different developmental stages that were tested in this experiment. And so what you see is that for effects on the mean, there are many cell types that are significantly decreased and as well as increased in these embryos. And we can also even find cell types that show increased variability at high temperature. And so this tells us something, I think, fairly significant, which is that after we account for stage in our model, so like I said, we can use position along this developmental stage trajectory as a covariate here. And so after we account for that, these embryos, we can see that they're actually missing certain cell types or have an altered composition. And just as a way of, I think, highlighting, why these computational methods are so powerful, or why this type of data structure is very powerful, I'm just going to highlight one of the cell types and how we followed up on it. And so that's the notochord. So the notochord showed a significant decrease in cell counts at high temperature, but also an increase in variability. And this was after accounting for the sort of mean variance relationship in all of these cell counts. So the notochord, maybe it's the defining feature of the chordates like us. And it's very important structurally during early development and also as a signaling center. So there are 2 main cell types in the notochord. There are these inflated vacuolated cells that you can see here. And then there's this epithelial sheath that are these cells around the outside that deposit this ECM sheath here, which you can kind of see in this electron micrograph. So if we extract just the notochord cells from the data set, again, now they're just colored by temperature from the reference. The high temperature cells are in here as well. We can distinguish these defining cell types. So the vacuolated cells, which are less abundant, are here, and the sheath cells that secrete this ECM are here. And using some spatial statistics, we found that the cells from high temperature embryos tended to occur among the sheath cells. So we think the cells that are responsible for this phenotype that we see in notochord cell abundance would be sheath cells rather than these vacuolated cells. So that's already one step in pinpointing the source of this phenotype. The other thing that's useful, of course, and I haven't really talked about this, is that there are transcriptional data underneath all of these results, so we can see what genes are expressed. And what we find in these high temperature sheath cells is that they show upregulated translation, so they're synthesizing a lot of protein, and they seem to have a high level of the unfolded protein response, which is a kind of stress response to deal with problems exactly like making too much protein. So it's a little bit paradoxical that this cell type is doing both of these things at once. They don't really go together. And so that was of interest to us. So we looked a little closer by sort of trying to understand what would happen in these cells if we took away its ability to manage protein folding stress by this unfolded protein response. And for that, we knocked out a key regulator of chaperones in the, sorry, I didn't say this, but this is an important response in the endoplasmic reticulum. So this is for protein synthesized in the ER. So we knocked out a regulator of these ER chaperones and then just looked at the fish. So at high temperature, of course, for wild type fish, we see this bent tail, as I mentioned. But if we take away this regulator, in this case, ATF6, we see that this phenotype is exacerbated, especially at high temperature, with lots of kinks. And maybe you can't see it so well, but this gray thing is the notochord. And these little kinks in it are not really common and also seem to lead to even more severe bends. And then when we look a little closer still, specifically to the sheath cells, we're able to do some TEM to look inside these cells and see that, so this is a sheath cell. This is the sheath that it secretes. What we found when we looked inside were collagen fibers that should be going here, but instead they're collecting inside the cell and they're not properly folded or processed. And we think this could be a consequence of this upregulation of protein synthesis to try to make more and more of this collagen that makes the sheath, but it's not actually working out. And at high temperature, the embryos can't actually manage to put it in the right place. And so we think this is important for causing this bent tail phenotype. All right, just gonna check the timing really quick. All right, so I guess I will wrap up. And so what I've shown you is that we have this new method to look at individual embryos with single-cell RNA-seq, and that kind of enables a new sort of phenotyping pipeline where we can actually pinpoint the effects of temperature, not just at the molecular level, but at the cell type or cell compositional level to identify what cell types might be responsible for phenotypes we see in these fish. And so I showed you that these specialized sheath cells in the notochord, these specialized sheath cells in the notochord had high levels of translation and of the unfolded protein response, and that the embryos as a whole had a high developmental rate. And so something we're looking into more and more now is the relationship between protein synthesis and this increased developmental rate. And just to show just a small result here, if we actually raise fish in the presence of a small amount of cyclohexamide to reduce levels of translation, but not really hurt the fish in any meaningful way, we can actually use this method to very rapidly stage these embryos and see that even at a temperature that should increase our developmental rate a lot, we can actually tone that down almost to wild type levels just by growing these fish in the presence of a translation inhibitor. So in that way, we're sort of trying to figure out more and more about this mechanistic link between protein synthesis or proteostasis and this acceleration of developmental rate. And lastly, I would just say a couple things about what my lab is continuing to work on here at EMBL. So we want to know whether we can predict this cell type specific temperature sensitivity. Are there other molecular features of notochord cells or of other cell types that showed reduced abundance at high temperature that would suggest their sensitivity even before doing this experiment? And secondly, we want to know whether any of these phenotypes that we see, any of this change in cell composition or the responses we see are adaptive in any way. And to that end, we're looking at a lot of different species, especially those that occupy different thermal regimes. And for this, we are looking for someone, postdoc in particular, to dig into this a little bit more deeply. And to end, before questions and all of that, I would just thank my lab here at EMBL, as well as several others back at UW Seattle, where I did a lot of this work, and thank all of you for your attention and would be happy to discuss.
Thank you very much. Right now, some time for questions. I come with you.
Thank you very much. That was really enlightening. I have a question to the correlation structure you showed, comparing the ages of cell types with other cell types within a fish. Did I see that correctly, that there was anti-correlation at 28 degrees, turning into positive correlation at higher temperatures? What cell types are those?
I actually didn't specifically follow up on what cell types those were. One thing I would be afraid of in terms of anti-correlations in the controls is that, there's a sort of problem here with annotating cell types and having a defined set of cell types. So it's possible that some of the cell types in the say 100 cell types that were in that matrix actually represent kind of more differentiated forms of the same lineage, where you might actually expect there to be anti-correlation, not just in their age, but even their abundance. And so, yeah, so I think this is something we need to account for and actually, I don't know, using that covariant structure, especially in abundance, to sort of inform us about like the lineage or the developmental structure of the animal might be able to even better isolate the effects of temperature. So I'm not sure the anti-correlation is a real thing, but I should look at the cell types.
Hi, pretty cool developmental biology talk. I really enjoy it. I have a question, sorry, regarding the notochord, that this is sensitive, sorry, to temperature. Concretely, if you can explain that of temperature sensitive state, like, can you give it an explanation like more in depth, please?
So the question is, what makes the notochord temperature sensitive, or did I?
No, sorry, what is the temperature specific state? I mean, how you assess that?
Oh, yeah. I mean, it was just the cells in that part of the UMAP. So we just use like a spatial statistic. Like, I think these had it was like basically a high level of clustering of the high temperature cells relative to the reference or 28 degree cells. So they were occupying that part of the map in an enriched way, if that makes sense.
Okay, thank you.
Yeah.
Hi. I was curious about the transfer label approach that you used, using the atlas. Is there a risk that you're kind of constraining yourself into what already is there in the healthy embryos? And like, if there's a new cell state that you never observe in healthy, how would the approach deal with that?
Yeah. My answer to this is that I personally am not so concerned, because I really, I've looked at the temperature data just without any of this projection and things like that. And I have to say, there aren't any new cell states. There are no new cell types. Really, all that happens is cell types get pushed, a little bit more or slower up and down their trajectories that already existed in the reference. However, in the sort of companion work for this, which I would encourage you to check out, which has lots and lots of mutants, I do think that's a much more relevant question, because you might actually see novel cell states. But even then, I think a lot of the things you see are stalled cell states or things that shouldn't be there at that time. But yeah, to answer your question, it is a real concern, and I sort of get around it by the fact that temperature isn't really affecting things enough to make a new cell state. But yeah, it certainly constrains what's available. Of course, you're using the model from the reference. So yeah. Yeah.
Hi, Michael. Thanks so much for the wonderful talk. My question is about the summarization that you did with the samples. I think it's really nice the way that you sort of have like fish and then the cell types. I mean, what you're showing is like an example of doing feature engineering on the single cell profiles, right? Because now you had genes, now you've got cells. Did you consider, or will you consider, other kinds of features like the mean expressions, variations, covariations, and so on?
Yeah, no, it's a really good question. I don't know that term, feature engineering, but it does sound nice. I'm glad that we're doing that.
That's what you do. Congrats.
Yeah, it certainly influenced a lot of our other analyses. So in terms of mean expression, we now do differential expression in terms of, we just basically bulk all of the cell types for each individual embryo and then do differential expression in that way. And actually, I like this a lot because we don't have to do tests on the individual cells, though I'm sure, there's good arguments to not do that. But yeah, it allows us to see the biological variation in a way that's, I think, really useful. But that's as far as I guess we've taken it. Yeah.
I guess maybe a little bit in the same direction as Sheila's question. I think the analysis sort of was good because it achieved a biological goal and sort of in a very straightforward and intuitive way. Now, having done it, where do you think would be the most fruitful avenues for further study, or maybe also new method development that maybe does this call here could be useful?
Yeah, I had mentioned before this kind of the cell type covariances. I mean, I think that's really, really important. So like one of the really obvious things you can see in the data is, muscle progenitors have an anti-correlation with the differentiated muscle cells. And that's very, very obvious and easy to understand. But the fact that you can do this systematically and look at across, many, many individual fish, I think opens up the opportunity to infer a kind of, if not lineage, some sort of at least hierarchy of cells, like what, if cell division in this progenitor affects the number of differentiated cells in the whole embryo, I think that signal is probably in this data somewhere. And I don't think we've done a great job of extracting that. Yeah, and, if you could infer something like lineage, at least in this one example you can, but if you could do it of the whole fish, this would be really, really powerful because, lineage tracing techniques are their own set of molecular tools that aren't that easy to implement. So yeah, that's one direction I think that would be really useful. And then beyond that, looking at variability in gene expression. So looking at genes that really do have higher variance. And I know that this is a little bit tricky because we're still working with single cell data, but I think when we bulk all of the cells from one individual fish, like I said, that's, for something like muscle, you have plenty of cells for that. I wouldn't do it for heart cells because you only get like 3 cells per fish. But for many of the cell types that are fairly abundant, I think you really could start looking at variability in gene expression as well.
Oh, sorry.
So thanks for a great talk. You didn't talk very much about sort of QA, QC type metrics, but it seems like there's a ton of variation. Each weld is sort of its own little batch, and the assumptions that you use to put everything together and do some of the transformations seem to sort of be somewhat elaborate. So can you just talk a little bit about how you thought about?
Yeah. Yeah. I mean, one thing we benefit from is with this Sky approach, we can actually process everything together. So a lot of the technical batch stuff, it's not really a strong signal. So you can, just co-embed these data without any of the batch correction. And they still, like all the cell types are still all the cell types. But for the reference, for example, it had so many time points that we had to generate it over the course of a few months. So we were constantly banking nuclei. And again, we got to pool all those nuclei in the end because that hash oligo, it basically comes in at the step of nuclear fixation. So when we process and dissociate the fish, we add that hash oligo, it goes into the freezer. And then when we finally pool all the samples together, we depend on that hash oligo to actually tell us, which sample is which. So we do actually strongly filter based on the frequency of basically one hash versus all of the other ones that you detect. So yeah, there's also a lot of, yes, there's a background distribution of these hash molecules. And so we have to set some kind of threshold to be conservative that we actually have, yeah, the right sample in that case. So we actually lose a lot of cells in that way. And that part could be optimized. And yeah.
I guess in particular, I'm wondering about edge effects because I have yet to encounter anything that has been run in plates that doesn't have edge effects.
Yeah, we didn't see. Yeah, so we looked at this for the basic QC metrics like number of UMIs per cell, number of cells per embryo. We didn't see a strong edge effect, but I'm with you. I mean, they're almost always edge effects. But yeah, I guess the dissociation happens fairly quickly. I mean, we could physically go through it. It's like, I mean, you're just going there with the multi-channel pipette, and I don't know. It might be useful to automate it in some way just to reduce, edge effects. But yeah, no, I guess. Oh, I see what you're saying. The fish actually, they aren't in the plate until dissociation. So they're in a more normal, homogeneous, like a petri dish, basically full of embryo media. And then it's only at the dissociation that they go into plates. So I agree. If we had actually grown up the fish for multiple days in the plates, you would have seen an effect. But this becomes too stressful because they run out of oxygen too quick. It's not enough water. And yeah, so especially at high temp. Thanks.
Thanks for again, so much to think about. I mean, also in terms of global warming and whatever. I mean, it's amazing.
And whatever. Yeah.
So then we move on to the pitch talks for the posters. Is that right? So very good.
Should we start it now? Okay. Yes, I will. Okay, so we're getting set up for the post pitch. All right. Okay. So we're going to be starting the poster pitches in about a minute or so, just getting the slides set up. So if you are one of the lucky ones who are going to be able to present, if you want to make your way over to this front area here.
On the future of science.
No. He's very happy with you.
So the Thursday just come after Wednesday, or how is the?
Yeah.
The slides.
It's after the shadow slide.
Right. Okay.
Nice. And microphone. Alrighty, thanks everyone for your patience. So we have our lightning round of presentations. I'd like to start by giving a round of applause to the brave souls who are going to be presenting. So you will be amazing. I will be very strict with timing, 1 minute. And if it goes over 1 minute, then we will all applaud you very nicely to thank you for your 1 minute of presentation. So without further ado, Ida, off you go.
Okay, so hello everybody. My name is Ida Holopainen, and I come from the Finnish Institute for Health and Welfare. And I have this poster presentation, which topic is means to visualize spatial patterns in host-associated microbiome data. So we have this like a nice data that is collected from a national pin risk study that combines health data with metagenomics. And we have linked the geographical information of the participants to study the spatial patterns of the gut microbiome data. And so the number of participants with both microbiome and spatial data is nearly 6,000, so which is quite a nice number. And the poster itself, it presents the visualization of our data and also 2 ways to visualize spatial patterns in microbiome data. So, I mean, please come and talk with me if you want to talk some visualizations of and spatial aspects of data. Thank you. See you.
Very good. Very nice. That was 56 seconds. Well done. All right. Next we have Philippe. Where is your microphone? Here we go. Try it.
Hello, hello. Yes.
All right, off you go.
Okay, so 3 keywords for my poster: flow cytometry, automated data analysis pipeline, and pre-processing and quality control. So regarding pre-processing and quality control, specifically for flow cytometry, it encompasses under the same umbrella a series of different steps, ranging from compensation to removal of different events of different nature. So if you want to implement this, soon you will be faced by a number of challenges. On the one hand, there is loads of possibilities in terms of which methods, which package you can use. Also within the methods, there might be loads of different parameter combinations, and even the steps themselves can vary. So the order of the steps and can have an impact on the results. And so on the other end, there is a lack of standardized tools, being it for pipeline design or for visualization and diagnostic. So in order to cope with those challenges, we developed the CytoPipeline, our package, which basically focus on 3 different parts, centralized and automated.
Thank you. Thanks very much. Well done. I gave you 5 seconds extra. All right, next person. Here we go. Next, we've got Caroline presenting. All right.
Yes, hello, I'm Caroline, and unlike many people here, I'm not a package developer. However, I have applied several Bioconductor packages to analyze a large bulk RNA sequencing data set with samples from 116 patients. And together with clinical and viability data, our aim is to understand drug perturbations, so the mode of action of different drugs, and also patient-specific pathway changes. And a special feature about this dataset is that we have used shallow 3' mRNA sequencing, which is a reliable but cost-efficient alternative to traditional sequencing. So if you're interested in how several Bioconductor packages can be applied to this type of data in order to gain mechanistic insights and information about pathway changes, I'm looking forward to see you at my poster.
Perfect. Well done. That was 57. Who will be the closest to the time? We will see. Next up, we've got Bartosz.
My name is Bartosz. I want to present you some solution for predicting function of unknown genes. Main reason why this tool was created is that each year number of sequencing genomes are growing, so does the number of unknown genes. To help researchers to narrow down possible function, possible option of function of a particular gene, we created this tool, and using protein domains as simple blocks of maybe some objects and some statistics, we are selecting which domain are significantly overexpressed in neighborhoods so they can probably share their function. Unfortunately, this tool is now written more in Python, but I'm trying to make some implementation for R, and this data, why it's in Python, this database is very large.
Excellent, very well done. Just got in within that grace period. Next, we have Ahmed.
Hi. So have you ever wondered how single cell methods commonly used in deal with immune genes that are hyperpolymorphic and polygenic? Well, the answer is simple. They don't. So this is problematic because this leads to erroneous quantification of important immune mediators and also impaired interdonor comparability, which ultimately obscures information immunological information contained in this data. So in our poster, we introduced the idea behind our new computational approach, which is to take into account those bioinformatic challenges that arise from immunogenetic diversity, simply by quantification immune gene expression at the allele, gene, and functional level. So if you're interested to learn more about this, please visit me at my poster. Thank you.
Excellent. That was 58 seconds, getting closer and closer. Next, we've got Reina presenting.
Hi, hello. I am Reina Cristina. I prefer saying Cristina, but my work is related with to identify immune response in marine organisms. But we are working with contamination. One concern contamination is benzoperin, and we use RNA-seq. But we had questions because we are no experts in bioinformatics. We wanted to know the difference using two different methods of packets to identify the differential gene expression. We use CODIF and DSEQ to determine this, using other analysis between like for biomarker, cluster profile, and G profile. Well, our results demonstrate very interesting things, and some are more similar, but others not. And this is the point. Well, this is very important for us to decide what biomarker or gene match with our thoughts of relevant question. And I please stop in my poster and give me advice. And I will tell you the final story about that.
Amazing. That was 1 minute exactly. Well done. Yes. All right, next we've got Anne-Catherine presenting.
Yeah, hi everybody. So quickly imagine the situation with me. You're writing your next paper about your enrichment analysis and you want to document all the necessary parameters to ensure that your analysis is really reproducible. And if you've been there, you know that's quite cumbersome. And we are proposing a solution for this because we know that enrichment methods matter, in short, EMMA. With our new framework, EMMA, you can use your usual enrichment method without changing much of it. But you get more than just the results. You get also all the necessary parameters to ensure a really nice documentation of your analysis and ensure your reproducibility. So if you want to get to know EMMA, please come visit our poster.
Excellent. And with like 20 seconds to spare. Well done. All right. Next, we've got Nasira presenting.
Hi, good afternoon. I'm Nasira, and I'm going to show you part of my analysis that we made to know the negative effects of benzoyperin over the time in the liver of the night tilapia exposed to benzoyperin. So we use the likelihood ratio test from the DESIC package. We also perform the enrichment analysis using cluster profiler for non-model organisms. And also we compare our results with the gProfiler platform. The result was very similar, but we are still exploring these results. And if you have any advice or a question about, you can welcome to my poster. So thank you.
Very good. And again, with 10 seconds to spare. Excellent.
Well,
all right. Bernard, you're jumping the gun.
I'll give you that.
Okay. Ready,
all right.
set.
Hello, everyone. So you should come see my poster. It outlines the work that I did, but also several other people, with the common goal of creating a corpus of standardized CiteSeq datasets and eventually performing automated annotation. Starting from the bottom, Song from the Fred Hutch Cancer Research Institute, together with me, have been working on several tools for performing phenotype discovery. That has been working on several tools and methods for performing protein normalization. Helen, who was supposed to come, but she was sick. She's been working very hard on a package for performing name standardization for proteins, and myself, that I've been trying to put everything together into a package, portable and reproducible, extensible pipeline. Thank you very much.
Amazing. Well done. And that was with 7 seconds to spare. So thank you very much to each and every presenter. One more. Please line up. And so that concludes the lightning round of the poster pitches. We are now heading into the poster session, as well as the GHGA sponsored coffee break. Now we are going to be back for the short talks at 4 p.m. Thanks very much.